Hi guys, welcome back to my channel. I'm so happy to see you all here. So this video is going to be fairly straightforward where I will be talking about 3 ways to convert Ensembl IDs to gene symbols. We all have dealt with gene lists at some point, and these gene lists have been derived from differentially expressed analysis, where we have a list of differentially expressed genes or transcripts, or the list of genes that are involved in a particular pathway. And oftentimes these genes are in different formats. They can be in the format of Ensembl ID or RefSec IDs or OnTrace IDs. And oftentimes we're required to convert the IDs from one format to the other. So there are various ways and methods available to do this conversion. But today I want to talk about 3 methods which we will utilize and which I will demonstrate as how can we use these methods to convert the ID from one format to the other. So for the first method, we will be utilizing Biomart. So Ensembl Biomart is a powerful web tool for performing complex querying and filtering of various Ensembl databases. It is often used for ID mappings and feature extraction. So today we will be utilizing both the web interface as well as the Biomart R package. The second method involves utilizing an R package called annotables. So the author of this package retrieved the data from the Biomart, cleaned up the column names, and saved the data as an R data package called annotables, which is accessible to all of us so that we can generate the gene ID mappings. And lastly, we will be utilizing AnnotationDbi R, which is also an R package that provides an interface for connecting and querying various annotation databases using SQLite data storage. Since we are dealing with Ensembl gene IDs today, I thought of talking about the Ensembl ID format. So an Ensembl ID is composed of 5 parts. The first part is the ENS, which tells us that it is an Ensembl ID. The second part is the species code, which is a 3-letter code. For human, there is no species code, but for other species like mus musculus, we have a 3-letter code like MUS. The third part is the object type, which tells us what the feature is that's associated with the ID. So we can have the feature could be either exon or gene or protein or transcript. Followed by that, we have identifier, a number that is associated with that object. And finally, after the decimal, we have the version number, which specifies the number of times the model has changed during its time in Ensembl. So we have an example Ensembl ID in the bottom left corner. So we start by ENS, which indicates it's an Ensembl ID, followed by MUS, which is a 3-letter code for mus musculus, followed by the letter G, which tells us that this ID is associated with the gene, followed by the identifier, and after the identifier and the decimal, we have the number 6, which tells us that it's on its 6th version. So for the tutorial today, we will be utilizing several R packages like Biomart, annotables, as well as organism specific database and Ensembl database. These 2 databases will be queried using the AnnotationDbi, and lastly, we will be using tidyverse to manipulate the data. So before we jump onto our studio, I will want to start the demonstration with Biomart web interface to show you how to find the gene symbols corresponding to the Ensembl gene IDs. So for the demonstration today, I have saved these 5 Ensembl gene IDs in a text file, and I want to find the corresponding gene symbols for these Ensembl IDs. In order to access the Biomart's web interface, we will be required to go on Ensembl genome browser website. So once we are on the genome browser website, we can see that Biomart is available in the tabs. So we click on Biomart, and this interface allows us essentially to build the query. So we start building the query by choosing the database, and the database that I will be choosing today is Ensembl genes. And once the database selection have been made, we choose the dataset. So there are a lot of datasets available, but I will be choosing human genes because the data that we have is associated with Homo sapiens. And once you make these selections, this query page automatically updates, giving you more options in the side panel. So now you will see you have access to filters and attributes. Filters can be used to define restrictions on your data. So let's say if you want to restrict your output to only contain genes that are present on a particular chromosome, let's say chromosome 3, then you can define those restrictions and filters using the filter parameters. whereas the attributes allows you to define the data that you want to retrieve. So let's say in addition to the gene ID and the gene symbol, you also want to retrieve the chromosomal coordinates. You can also do that by making those selections in the attributes. Let us start by going to filters, and as I said, you can add restrictions to your data. So there are a lot of ways you can restrict your data, like you can define certain regions, as I said, genes, phenotypes, gene ontology terms, and a lot of other options. I will be going in the gene panel because I want to restrict my search only to those IDs that I input, and I will be checking this box because we have the Ensembl gene IDs. And again, you can input the data in various formats. It accepts a lot of formats here, so you can take a look at what data you have in what format, and you can accordingly convert and get the corresponding gene symbols. So there are two ways: either you can paste those gene IDs, or you can upload a file. So I'm going to upload a file by clicking on choose file, and this is my file, so I'm going to upload it. Now that I've restricted my search to only those gene IDs present in my file, I want to go to attributes to define what data I want to retrieve. So I wish to retrieve not only the gene symbols but also the gene IDs that I originally inputted in my text file, because I want to see the mappings. I do not wish to see the transcript IDs, so I will uncheck these boxes and I will check the box containing the gene name. There are also additional information you can retrieve corresponding to your gene IDs and symbols, but at this point I do not wish to have any more information except for the gene ID to symbol mapping. Now that we have made our selections, let's go to result button. And when we click on the result button, we get a table with the data that we have chosen in our attributes. So we have the gene stable IDs. This was also our input, and we got the corresponding gene name. In addition, we had also chosen to have the gene stable ID version. So this is the gene IDs with the version information as well. So this data can be downloaded by clicking on the export all results to. You can select the drop down. You can select whether you want the file to be compressed. You can select various formats you want to, the various formats that are available for your data to be downloaded in, and then you can click on go to download these results. We utilize Biomart's web interface to get the gene ID mappings with the gene symbol. Now let's repeat the same process utilizing Biomart's R package and performing those steps programmatically. In addition, I also want to demonstrate how to utilize other packages to get the gene ID mappings. Let's start by reading our libraries. After reading our libraries, we will read in the Ensembl gene IDs. Let's save that to a variable. So here are our 5 Ensembl IDs. The first thing we did on Biomart's web interface was to choose the dataset. So we do the same thing here, and in order to get the list of available dataset, we run a function called list ensemble. List ensemble shows us the available databases as well as the values for those databases. So if you want to use, let's say, database Ensembl genes 105, then we need to use values genes to indicate that we are intending to use this database. So let us select the database. So we use the function use ensemble, and the first parameter is Biomart, where we provide this value which indicates what database we want to use. And let's save this in a variable called Ensembl. After choosing the database, the next thing we need to know is the dataset that we want to use. So we use a function called list datasets and use Ensembl here as the parameter, and save this to a variable called datasets. Let's take a look at this datasets, and we see that there are around 214 datasets available. So the dataset that we are interested in using is from humans. So I'm quickly searching that in the search bar, and this is the dataset that corresponds to the human genes. So I know that this is the dataset that I want to use. Now that we know the dataset and the database that we want to use, let's connect them using useMart function, and the first parameter would be the name of the database. We have saved that in Ensembl, and the next parameter is dataset, and that is the name of the dataset, which is from the datasets variable. So I'm just going to copy paste the value, and let's save this in a new variable called Ensembl connection. This has to be in the string, in the quotes actually. Now that we have connected the database and the data set, let us build the query now. So to build the query, we use getBM function, which has various parameters like attributes, filters, values, and mart. Attributes, as I previously mentioned, are used to define what data you want to retrieve. So this will be a character vector of all the values that you want to retrieve. Filters are used to put restrictions on the data that you want to retrieve. So it will be a vector of filters that you will use as input to the query. Values would be the values for the filters, and this will be more clear when we actually build the query. And the mart here will be our Ensembl connection object. So to define what data we need to retrieve, we need to get the attributes. So there is a function called list attributes, and we provide the Ensembl connection object as parameter and save this to attributes variable. So there are around 3143 attributes available. So the first thing we need to retrieve is the gene name. So the gene name corresponds to the value external gene name. So this will be one of the value in our attribute character vector. The next thing we also want to retrieve is in addition to the gene names or the gene symbols, we also want to retrieve the original gene IDs that we are inputting. So that is gene stable ID, and gene stable ID corresponds to value Ensembl gene ID. So we will be using these two values, the Ensembl gene ID and the external gene name, as the values in our vector, which will be provided to the attributes. So we define a character vector, and we want external gene name in addition to Ensembl gene ID. I'm just going to copy paste it here. So instead of putting it after the external gene name, I want to put it before. So the attributes that we want to retrieve are the Ensembl gene ID, the IDs that we input, and the corresponding external gene name, which corresponds to the gene symbols. Similarly, we want to retrieve the filters, and the filter here would be, we are inputting a list of Ensembl gene IDs. So we need to get the filter value that corresponds to the Ensembl gene stable ID. So similarly, we can get list filters provided with the Ensembl .connection object as the parameter and save it to a variable called filters. So we have around 439 filters available, and as I said, I want the filter to be gene stable ID, and that corresponds to Ensembl gene ID. So I will copy the name of the filter and paste it here. Values would be the values for the filter that we defined. So we asked Biomart to restrict searching for gene symbols only corresponding to the gene IDs that we provide. So in the values parameter, we actually provide the values for the gene IDs, the input gene IDs, which is Ensembl IDs and the column V1, because our gene IDs are present in Ensembl IDs, data frame, and column V1. We have finished building a query, and now let's run this. It seems that the Ensembl.connection will not be in quotes. And now you can see that for the input gene IDs, the Ensembl gene IDs, we have the corresponding gene symbols. Moving on to the next method using annotables. In the annotables package, the annotation data is available for various species, and for each species we can access the table using the genome build. So for human, we can access either GRCH37, which is HG19, or GRCH38, which is the latest build, and we can directly access it as a table. So before I do anything, I just want to show you the table. So when you simply run grch38, you can get the table that has all the information corresponding to the gene IDs, the gene symbols, the entrees IDs, the chromosome coordinates. So let us filter this to only contain those genes that are present in our gene IDs. So we can filter this table, and we want only those gene IDs that are in our Ensembl IDs data frame in column V1. And when we run this, we get all the information that's corresponding to the gene IDs that are present in our list, and we have all the corresponding information. Finally, we come to the third method, which utilizes AnnotationDbi's functions to query databases. So we have organism-specific database and Ensembl database. We will query those databases using the AnnotationDbi's function to retrieve the gene ID to gene symbol mappings. So we use a function called map IDs. And the first parameter here will be the name of the database. And for us, it is the organism specific database, which is the first database that we would want to query. Following that, we have some parameters we need to define, which is keys. We have key type, and lastly column. So this is not quite different from what we did with the Biomart. So the keys here correspond to the values in Biomart. So we have to provide the input gene IDs for which we need the corresponding gene symbols, and we provide it as the vector here. Next, we have to define what the key type is. So we need to define what information are we providing, what is the type of that information. So for that, we can look up what are the key types available for this database. So we're just going to copy the name of the database and paste it here. So when you run the key types with the name of the database, you get all these key types available. So the input keys are in the Ensembl ID format, and so essentially our key type would be Ensembl. And we can also look for the columns which are available to retrieve. So the column here correspond to attributes, where key type correspond to the filters in Biomart format. So we do the same for the columns, and we find that these are the columns available to retrieve, and what we're interested is in the gene symbol. So essentially we'll copy the name symbol, and this is the data that we're interested to retrieve. So now that we have built our query, now let's run this, and we can see that for every gene ID that we provided, we have a corresponding gene symbol being retrieved. Let us repeat the same process for the other database, the Ensembl database. So we have map IDs. This is the function from the AnnotationDbi, and the database that we want to use is the Ensembl DB. And similarly, we define keys, we define key type, and we define column. So the keys here will remain the same. We are providing the input as Ensembl gene IDs. We provide it as a vector. The key type, which corresponds to the filter, which is analogous to filters in the Biomart build query. So for that, we will look up the key types that are available in the Ensembl database. And since our input is the gene ID, here we do not have the Ensembl, but we have gene ID. So we will be copying and pasting gene ID. And we will look up the columns information for the same database. And similarly, we are interested in retrieving the symbol. So we will copy symbol and paste it here. And this follows the exact same format, just that the key type information has been different for both the databases. It varies with the database, so you might have to check it before you run it. And once we have built the query, we run this, and similarly, like the previous database, we also retrieved a named list of the gene IDs with their corresponding gene symbol. So these were the three methods to convert Ensembl IDs to gene symbols. I'm sure there are many other methods, but I chose these three methods as these are the methods that I frequently use. As always, I will upload my script to the GitHub, and I will link my GitHub repository in the description below. So if you found this video helpful and informative, please make sure you hit the subscribe button, like the video, share it, and leave your comments under the comment section. Until next time, see you.